genetic engineering. Okay, so today I want to discuss about DNA cloning. DNA cloning is used to create copies of a particular gene of interest. So literally, clone means a collection of molecules or cells all identical to an original molecule or cell is called a clone. So clone is genetically identical organisms or molecules derived from a common ancestor is called clone. Okay, so generally if you see plant clone, generally we refer in plants propagation, clonal propagation, micropropagation. Okay, so if you want to clone plants, so plants have the ability, totipotency. So means every plant cell able to produce an entire plant. Every plant cell have the ability to produce an entire plant. This property is called totipotency. So based on that property, you can produce so many identical plants. Okay, so in the tissue culture, in the third semester, you are going to study plant tissue culture. So in the plant tissue culture, you can take it few cells or tissues from the plant, and you can culture on artificial media supplemented with macroelements, microelements, vitamins, and growth hormones, and a carbon source under controlled conditions. Okay, so if you inoculate a tissue in aseptic conditions, the tissue will divide into callus. Later, the callus developed into an entire plantlet. Okay, na? So in that way, you can produce clones or identical plants. Okay, so generally cloning refers to plants. So coming to the DNA cloning, so the DNA cloning can be defined as a method for identifying and purifying a particular DNA fragment of interest from a complex mixture of DNA fragments, and then producing large number of the fragment of interest is called DNA cloning. And gene cloning means when DNA is extracted from an organism, all its genes are obtained. In gene DNA cloning, a particular gene is copied. Then why DNA cloning? So with for genetic engineering and for rDNA technology, we need a DNA cloning. So not only for the genetic engineering purpose or rDNA purpose, the cloning of DNA can also be useful for many aspects. So a particular gene can be isolated and its nucleotide sequence determined. Control sequences of DNA can be identified and analyzed. Protein, enzyme, or RNA function can be investigated. Mutations can be identified with using DNA cloning. And also, organisms can be engineered for specific purposes. So, such as in medical biotechnology, we can produce medical treatment proteins, vaccines, biopharmaceuticals, example, insulin, and also can be used in the genetic engineering in crop improvement. So developing insect-resistant plants, pest-resistant plants. To overcome the abiotic and biotic stress factors, okay, we need DNA clonings. Okay, na? Then sources of DNA for cloning. So we can take DNA from any source. We can take DNA from the microorganism or plants or from animals. And with using genetic engineering, you can transfer DNA from plants to the animals, animals to the plants, microorganism to the plants, and plants to the microorganism. Okay, and the source of DNA maybe it's you can take it from chromosomal DNA or extra chromosomal DNA, or you can take cDNA, RNA converted to cDNA, by the way of with using reverse transcriptase. And also PCR amplified DNA can also be taken as source for DNA cloning. And these are the steps in cloning a single piece of DNA. Okay, so there are 10 steps that involved in the cloning of DNA. One is isolation and selection of DNA and identification of appropriate restriction sites. Second is cut vector and desirable DNA with restriction endonucleases. And next step is run on gel to separate fragments. So that is an agarose-gel electrophoresis. And isolate specific fragment. And the ligation with the cut vector. Next step is transform host bacteria. Next step is selection of DNA clones and their expression and grow up colonies. And the 8th step is isolate plasmid DNA. And 9th step is cut with restriction enzymes to confirm presence of foreign DNA. And the last step is run on gel to identify recombinant plasmids. Okay, so these are the steps that involved in the cloning of DNA. Here you can see representation of cloning of DNA. The first step is selection of DNA. And the second step is cut DNA with restriction enzymes and mix with cut vector and ligase enzyme. And later, after this vector DNA cloning, it's also called construct. This constructed vector can be introduced into the bacteria. Okay, thereby this desirable DNA will be expressed in that bacteria. Okay, so later you can play it on agar with antibiotic selection. Okay, so antibiotic resistant genes can be incorporated at the time of DNA cloning. Okay, for the selection purposes. Okay, so in the DNA cloning, so while constructing the vectors, the points you have to consider while making the plasmid or vector along with the desirable DNA, you have to tag your desirable DNA with selection genes. For the selection purpose, you have to use marker genes and reporter genes. Okay, so marker genes and reporter genes are the selectable genes. You have to tag these genes along with your desirable DNA in the vector constructioning for the selection of RDNA clones after transformation. Because all these experiments are molecular level, you cannot observe the things with the naked eye. Okay, na? But if you use a restriction with marker genes and reporter genes, these marker genes and reporter genes helpful to select whether transformation successful or not, and also the presence of reporter gene and marker gene indicates the presence of desirable gene. Okay, na? So because you are adding your marker genes and reporter genes along with desirable gene, and after transformation, when you play it onto the agar media, your the media, due to the presence of marker genes and reporter genes, okay, so suppose if you see a Gus gene, that will give color, like GFB gene, that will give color. So based on, by looking that color, you can automatically confirms the presence of desirable DNA. Okay, so these are some of the features of cloning of DNA, and in this there are so many tools we have to use for the cloning of DNA. The first tool is restriction endonuclease. Okay, so for DNA cloning, we need so many enzymes and machinery. Okay, so the first tool is restriction endonucleases. Then, what is restriction and what are restriction engines? Restriction engines are, these are the engines that cut DNA at specific sites. They are properly called restriction endonucleases because they cut the bonds in the middle of the polynucleotide chain. Some restriction engines cut straight across both chains, forming blunt ends, but most engines make a staggered cut in the two strands, forming sticky ends. The cut ends are sticky because they have short stretches of single-stranded DNA with complementary sequences. These sticky ends will stick to another piece of DNA by complementary base pairing, but only if they have both been cut with the same restriction enzyme. Restriction enzymes are highly specific and will only cut DNA at a specific base sequence. They are about 4 to 8 base pairing long, and they called recognition sequences. Restriction engines are produced naturally by bacteria as a defense against viruses. They restrict viral growth, but they are enormously useful in genetic engineering for cutting DNA at precise places. Also, these restriction engines are also called molecular scissors, and these restriction engines are short lengths of DNA cut out by restriction engines are called restriction fragments. There are thousands of different restriction engines now with over 100 different recognition sequences. Restriction engines are named after the bacteria species they came from. So I will tell you the nomenclature of restriction engines, and there you can find why they are called this EcoR1 and HIND3 like that. Okay, so the nomenclature of restriction endonucleases and the properties. So nomenclature: Smith and Nathan's proposed enzyme naming scheme. 3-letter acronym for each enzyme derived from the source organism. So if you see this HIND3, HIND2, first letter from genus, and next 2 letters represent the species. Here you can see in. Additional letter or number represent the strain or serotypes. Okay, here if you see HIND2, so for example, the enzyme HIND2 was isolated from Haemophilus influenzae serotype D. Okay, so why? So these are the points you have to consider for nomenclature, and there are so many engines are there. Here I am showing few restriction engines: BAM-H1, EcoR1, HIND3, MBO1, PST1, SME1, TAC1, XMA1. These are some of the restriction engines now we are using for the genetic engineering and rDNA purpose. Okay, na? So here you can see BAM-H1 isolated from Bacillus amyloliquefans. Okay, and EcoR1 are isolated from the Escherichia coli RY13. Like that, HIND3 or Haemophilus influenzae. Okay, so like that, MBO and all these. Okay, so SMA, Serratia marcescens. Okay, so these are the some of the restriction endonucleases. And classification. You can write these examples. And classification of restriction endonucleases. Synonyms to restriction endonucleases are also called endonucleases because cut DNA from inside. All, if you see, all restriction endonucleases are highly heterogeneous, evolved independently rather than diverging from a common ancestor, and broadly classified into 4 types of restriction enzymes. Okay. RM system, restriction modification system. Endonuclease activity cuts foreign DNA at the recognition site. Methyl transferase activity protects host DNA from cleavage by the restriction enzyme. Methylate one of the bases in each strand. Restriction enzyme and its cognate modification system constitute the RM system. And production of self-DNA. Bacteria protect their self-DNA from restriction digestion by the methylation of its recognition sites. So methylation is nothing but the addition of CH3 group to the DNA. Restriction enzymes are classified based on the recognition sequences and methylation patterns. And there are 4 classes of restriction enzymes are there. They are type 1, type 3, type 4, and type 2 enzymes. Okay, most widely used enzymes are type 2 enzymes. So type 1 enzymes are multi-subunit protein enzymes, function as a single protein complex containing 2 restriction subunits, 2 methylation subunits, and 1 specificity subunit. Cleave DNA at a random length from recognition site. These are the properties of type 1. Okay. Are you following?
Yes, sir.
Are you writing points?
Yes.
So please note down on your running notes, because I am compiling all these slides from different books. Okay. So please note down each and every slide. Note down all points. Okay. Can I go for the next slide?
Yes, sir.
Okay. Type 3 enzymes. Okay, I will explain type 2 later, because type 2 enzymes are the most widely used enzymes in the genetic engineering process.
The type 2, sir.
Type 2 later. Okay, so first I want to explain type 3 and type 4. Later I will explain type 2. Okay, so type 3 properties, if you see the properties of type 3, they are large enzymes, combination, restriction, and modification enzymes, cleave outside of their recognition sequences, require 2 recognition sequences in opposite orientation within the same DNA molecule. No commercial use or availability. Okay, so type 3, we cannot use it for commercial purpose. And type 4 are cleave only modified DNA. So either methylated, either hydroxymethylated, or glucosyl hydroxymethylated bases only. Recognition sequences have not been well defined. Cleavage takes place nearly 30 base pairs away from one of the sites. Sequence similarity suggests many such systems in other bacteria and archaea. These are the properties of type 4 restriction enzymes. Okay, and coming to the type 2 enzymes. So these type 2 enzymes are very important. Now we are using about 3,500 restriction enzymes in genetic engineering purpose. Type 2 enzymes are the most useful for gene analysis and cloning. If you see the properties of type 2, They recognize 4 to 8 base pair sequences. They need a magnesium 2 plus as cofactor, cut in close proximity of the recognition site, homodimers, and ATP hydrolysis is not required for the type 2 digestion. So these are the properties of type 2 restriction endonucleases. Okay, please note down. These restriction engines that not only useful for the genetic engineering purpose, but also be used in the DNA fingerprinting. Okay, so you have RFLP method, restriction fragment length polymorphism. So in the RFLP, it's one type of DNA fingerprinting method. With using RFLP, we can identify crimes, okay, and evolution. We can study evolution we can use it for evolution studies, and parental, you can set parental disputes and all. Okay, so anyhow, in the coming classes you will study about DNA fingerprinting also. So recognition sequences, each restriction enzyme allows cuts at the same recognition sequences, produce the same gel banding pattern. Many restriction sequences are palindromic. So example, you can read it, read the same in the opposite direction, like madam, race car, etc. Okay, and these restriction engines, here you need to define two terms, blunt ends and sticky ends. So after applying restriction endonucleases, a straight cut of restriction enzymes generate blunt ends where both strands terminate in a base pair. Blunt ends are also called non-cohesive ends since there is no unpaired DNA strand fleeting at the end of DNA. Okay, so blunt ends, some restriction enzyme cut DNA at opposite base. They leave blunt-ended DNA fragments. These blunt-ended fragments can be joined to any other DNA fragment with blunt ends. Enzyme useful for certain types of DNA cloning experiments. Okay, so these are the blunt ends that gives two fragments. Okay, and next sticky ends. Okay, so most restriction engines make staggered cuts. Staggered cuts produce single-stranded sticky ends. Okay, here you can see the restriction enzyme cleaving point. Okay, in this manner, it cuts the DNA and that generates sticky ends. Okay, so these type of sticky ends are useful in the genetic engineering experiments and for the ordinary technology. Here you can see you have ALU1 and HAE3. If you apply ALU1 and HAE3, they produce blunt ends. And if you use BAM-H1 and HIND3 and EcoR1, they produce sticky ends. Okay. So in the genetic engineering process, mostly we use EcoR1, HIND3 and BAM-H1 because they generate sticky ends. Here you can see if you apply ALU1 gene that comes like this, and if you apply BAM-H1, HIND3 and EcoR1, they cuts at a specific sequence and they generate sticky ends. These are helpful in cloning of DNA and construction of vector. Please note down the slide. So restriction engines helpful in DNA fingerprinting, and that is called RFLP and restriction mapping. So restriction mapping is the process of obtaining structural information on the piece of DNA by the use of restriction engines. Restriction. So if you see the restriction mapping, restriction mapping involves digesting DNA with a series of restriction engines and then separating the resultant DNA fragments by agarose electrophorese. The distance between restriction engine sites can be determined by the patterns of fragments that are produced by the restriction enzyme digestion. Here you can see in this slide, you can see the restriction mapping. So with using restriction endonucleases, you can use this system in the DNA fingerprinting system. Here you can see this. After digestion, you have to run that samples on the agarose electrophoresis. Okay, here this is the marker line suspect to skin and nails of the victim. Okay, hair found in the criminal site, and victim. Okay, control and marker and control DNA. Here you can see. So based on this, you can report. So this is called DNA fingerprinting. Okay, so this is about restriction endonucleases.